we'll start off with me giving a pre-lab lecture like I normally do, and then you're going to set up some restriction digestions on those on the plasmid DNA that you isolated last week. And then the digestion is going to go on for, I don't know, about an hour or so. And I guess what I'm warning you is there's about an hour of downtime in the lab today, so if you want to bring study materials for this class or other class, that might be a good thing to bring to laboratory today. What else was I going to tell you about? The Friday quiz in here, like 2 days from now, is going to be just on this chapter we're covering now, genome, so be ready to answer questions on that. I guess that's it. Well, we don't have too many lectures left. We've got today's lecture and Friday's lecture, and then next week, Monday's lecture and Wednesday lecture, because boy, when is our final in here? It's yeah, next Friday. Yeah, okay, that's right. There is no lecture here next Friday because the final exam is on that day. Yeah, Friday, May 25th. The final exam is at 11:30. For some reason, they don't schedule them at the normal meeting time of the class. Anyway, so next Friday, the 25th, 11:30, in here, in this room, not in the lab room, is the final exam. Any final exam related questions? All right. Well, let's see. We are on this chapter on genomes, and we've been focusing on differential gene expression, which, remember, is how each cell can express only some of the genes that's in its genome, because all your cells have all 25,000 genes, but they typically at any given time are only expressing about 1/5 of those. So people wonder, well, how can cells turn on some genes and turn off other genes? And one of the major points of this is that the most differential gene expression, the most regulation of gene expression takes place here at the transcriptional level. If the cell wants to express the gene, it makes the mRNA, and if it doesn't want to express the gene, it doesn't make the mRNA. And in the last couple lectures, we talked about how the cells can regulate transcription of the gene. In eukaryotes, there were things called activator proteins, which bind to some DNA sequences called control elements or enhancers. And these activator proteins, once they bind to the control elements of the gene, help the RNA polymerase II attach to the template strand. And, well, that's how differential gene expression works. If a cell wants to express a gene, then the cell has the activator proteins that bind to that gene's control elements. And if a cell doesn't want to express a gene, it does not have those activator proteins for binding to that gene's control elements. And there were also things called repressor proteins, which in some cases just simply blocked the activator proteins. Repressor proteins, at least some repressor proteins, bind to the same control elements, but when they do, they physically block the activator proteins from attaching, so that could help shut off a gene. There was a couple other ways that repressor proteins can work. There we go. Some repressor proteins, instead of binding to the control elements of the gene, some repressor proteins instead modify the histones around the gene. By changing the histones, they could cause the DNA in that area of the chromosome to wrap more tightly around the histones. And when the DNA wraps tightly around the histones, there's just no room for the activator proteins and the transcription factors and the RNA polymerase II enzyme to get in there, so you tend to decrease the amount of transcription from the gene. Gesundheit. And there's other types of repressor proteins that do this. They go to where the gene is, and they add methyl groups, CH3 groups, to the actual DNA, to the G's and A's and T's and C's. And as it turns out, methylating the DNA also inhibits transcription. It just must somehow get in the way of the activator proteins in the RNA polymerase too. So those types of things, DNA methylation and histone modification, they both decrease transcription of the gene, but by actually modifying the DNA, or at least modifying the chromatin, which is the DNA, as it's wrapped around the histones.
When the repressor proteins modify the histones, how do they do that?
They add something called an acetyl group to the histones, and or I should say that they actually modify, they actually remove the acetyl groups. These little purple things I think are supposed to be the acetyl groups on the histones. And if the acetyl groups are present, that tends to push the DNA away, and so you get a more spread out chromatin. And if the acetyl groups are removed, then that tends to attract the DNA more, and the DNA wraps more tightly around the histones. All right. Let's see. Oh yeah, there was a review concept I was going to put out here. You also find activator proteins and repressor proteins in prokaryotes, but the difference is where they bind. If this is the promoter, and here are the actual codons, in prokaryotes, the repressor proteins and the activator proteins both bind to the promoter region itself and can either block or assist the RNA polymerase from binding to the promoter region. But in eukaryotes, remember, way far away from the promoter are the control elements. And the activator proteins and the repressor proteins bind there on the control elements, not in the promoter region. Okay, well, let's see. Most of the differential gene expression, it takes place in regulating transcription right there. But the point I think I was making at the end of the last lecture was this: although that's where most of the differential gene expression takes place, there's lots of other steps where it can, and in some cases does, take place. For instance, when the transcription takes place, the initial thing you get is called the primary transcript. And remember, the primary transcript needs to be processed before it becomes a mature mRNA. Introns have to be removed, poly A tail has to be added, the 5' cap has to be added, that sort of thing. And so you could regulate that step to affect gene expression. And then the mRNA exits the nucleus and goes out to the cytoplasm, and then the ribosome has to bind onto the mRNA and scan along it looking for the start codon. And then once it finds the start codon, the ribosome has to assemble all the amino acids. There are ways to block that step, so you can regulate gene expression there. There are enzymes in the cytoplasm that can degrade mRNAs. That's another opportunity to regulate gene expression by regulating the lifespan of an RNA, a particular RNA. And even after a protein has been successfully manufactured by the ribosome, there are ways of regulating the activity of that protein, ways of increasing its activity or decreasing its activity. And so that can also be included as differential gene expression, in the sense that it can control how much or how little of the active form of the protein you have in the cell. So I thought I would look a little closer at some of these for the idea that you can regulate gene expression here at the processing of the primary transcript. What that mostly involves is the splicing of the introns and exons. Recall that the primary transcript has some regions that are called the exons. They're the colorful boxes there that actually have the codons that encode the actual amino acids. But the exons are separated by regions called introns, the tan-colored areas here, that don't encode any protein. I think I said that think of those as like the advertisements in a magazine that you're reading. You don't really care about those, and everything else being equal, you'd wish they were gone. And so that's what happens inside the nucleus. The intron sequences are spliced out, and there are, remember, there's some enzymes in the nucleus called SNRPs that bind to the exon-intron boundaries, like there and there and there and there and there and there, and then the SNRPs splice out the introns and ligate together the exons, and when all the SNRPs cluster together, it's called a spliceosome. And in any event, the exact splicing pattern that the SNRPs and the spliceosomes do can vary. In some cases, you might find something like this, where this exon gets spliced to this exon right there, and this whole thing, including this exon right here, gets cut out. So you would get one type of protein out of that, whereas in other cases, the SNRPs could splice like this, where they cut out this exon and instead use this exon right there. So you get a different protein coming from that messenger RNA. And that's called alternative splicing of the primary transcript, or the pre-mRNA. And since it can lead to different proteins, I mean, the mRNA right here, the protein that gets made from this one is going to be different from the protein that gets made from that one. That is a type of differential gene expression. And I believe they haven't fully figured out yet how the cell regulates the SNRPs in telling them which exons to cut out and which exons to include. But in some way, there must be some enzymes that control that process, that tell the SNRPs where to splice. And so by varying those enzymes, the cell can do differential gene expression at the pre-mRNA splicing. All right, well, let's go to the next step. Let's say that these mature mRNAs made it out into the cytoplasm. Here they are. Once a mRNA makes it out into the cytoplasm, the ribosome has to bind onto it and then scan along it looking for the start codon, and once it finds the start codon, the ribosome starts piecing together the actual protein. What they found is that in the cytoplasm, there are some proteins that can bind on to the messenger RNA and that serve to block the ribosome, to stop the ribosome from scanning along. And then that would end up, well, if the ribosome is blocked, it's not going to be able to make the complete protein, and that's a way of downregulating the expression of that protein, gesundheit. And here's a cartoon of that. There's the messenger RNA, and here comes one of these blocking proteins. It attaches to the mRNA, and then here comes the ribosome to try to scan along and look for the start codon, but crash, it runs into the blocking protein, and so it's never going to be able to initiate translation, or even if it does manage to start a few amino acids of the translation, it won't be able to finish the protein. There are other proteins they find in the cytoplasm that bind to the messenger RNAs and then degrade them. They have, there's an enzymatic activity here that can chop up the messenger RNA. I think I might have talked about these ones before. You may say, well, why would the cell want to cut up its own messenger mRNAs? Well, just because the cell makes a messenger RNA for a protein at some point in time, the cell may not necessarily always want to have that protein around. And so these RNA degrading enzymes are just a way of having a finite lifespan on any particular mRNA, just so that this mRNA doesn't always hang around and be getting proteins made from it. So these guys just float around attaching to messenger RNAs and chop them up. I'm not sure exactly how many minutes any given mRNA survives in the cytoplasm. I really don't know, but if I had to take a guess, maybe only 5 minutes or so before it's degraded. Okay, now these two things that I just mentioned, these proteins in the cytoplasm that can attach onto an mRNA and block the ribosome from translating it, and these other enzymes that can attach onto an mRNA and degrade that mRNA. As I've described them so far, I didn't mention any way that they could target any specific mRNA. You know, just in and of themselves, it seems like they could either downregulate the expression of all the mRNAs. But this concept of differential gene expression centers on the idea that the cell might be able to decrease the expression of certain genes, but not of other genes. And so we might ask ourselves, well, is there a way for the cell to target only certain mRNAs to be blocked from translation? Or can the cell target only certain mRNAs to be degraded by these RNA degrading enzymes? And the answer is yes. Here's another messenger RNA. And if you think about it, what makes an mRNA different from another mRNA, one mRNA different from another, is the sequence, because different mRNAs come from different genes and they encode different proteins, and so therefore they're going to have different sequences of the G's and A's and U's and C's throughout the messenger RNA. And so by keying in on differences in the mRNA sequence, the cell is able to selectively block translation of only certain mRNAs